This 3D rendering isolates the position of the kidneys within the human torso. Positioned just beneath the rib cage and nestled against the posterior wall, these two bean-shaped organs are wired directly into the central vascular system. These organs function as active regulators, constantly scanning and adjusting the chemical concentrations of the blood to keep the body's internal environment within a narrow, viable range. Every second, human metabolism generates a continuous byproduct of nitrogenous wastes, specifically urea and uric acid. If these compounds are allowed to accumulate in the bloodstream, the resulting toxicity is rapidly fatal. To clear those toxins, the body must flush them out using water. However, total bodily fluid volume and specific electrolyte concentrations must remain stable to support cardiac and neurological function. Sustaining this balance requires a system that can selectively retain critical nutrients while simultaneously purging metabolic toxins. The biological solution to this dual challenge relies on massive parallel processing. Instead of routing blood through a single large filtration membrane, the kidney distributes the workload across a network of approximately 1 million microscopic, independent functional units. To understand how chemical stability is maintained, we must trace the exact chronological pathway of fluid as it navigates this architectural labyrinth. The macro-level connections dictate the bulk transfer. High-pressure, unfiltered blood enters via the renal artery, and processed blood returns to the systemic circulation through the renal vein. The true complexity of the renal system lies in the internal, sequential fluid routing logic trapped between that entry and exit. Moving past the exterior surface reveals a strict spatial geography designed to organize and distribute that incoming fluid. This schematic shows the internal cross-section. The indentation is the hilum. The red renal artery enters here. Pressurized blood forces through, brighting outward, contained by the thin outer perimeter, the renal capsule. Immediately beneath sits the renal cortex, a wide ring of active tissue. Because of the outward branching of the arterial network, the cortex serves as the primary destination for the raw blood supply. It operates as the staging ground for the initial high-pressure processing stage. Beneath the cortex lies the deep inner core of the organ, known as the renal medulla. The medulla is organized into distinct cone-shaped geometries known as renal pyramids. This macroarchitecture dictates a physical sequence. The organ routes raw fluid outward to the perimeter for initial sorting, and then channels the resulting liquid waste inward toward the core for elimination. Zooming closely into the dense outer tissue of the renal cortex reveals the exact microscopic machinery performing this incredible extraction process. This schematic isolates a single nephron, the fundamental microscopic functional unit. The red arterial supply line narrows into the afferent arteriole. This narrowing increases internal fluid pressure, forcing the high-pressure stream into the glomerulus, a dense knot of porous capillaries. Glomerular filtration is a mechanical process. The extreme internal pressure forces materials through a microscopic biological mesh. A physical separation occurs. Water and nitrogenous wastes are pushed out, while large red blood cells are retained. The extracted material is captured by Bowman's capsule, a spherical funnel tightly enveloping the capillary knot. Remaining cellular blood exits via the efferent arteriole. The glomerulus operates as a blunt force separator, dividing bulk plasma from the essential cellular architecture of the blood. Once the cellular blood is separated, the mechanical focus shifts to the extracted filtrate, now isolated in Bowman's capsule. This filtrate enters the first stage of the tubular labyrinth. This twisting section adjacent to the capsule is the proximal convoluted tubule, or PCT. The PCT functions as a reclamation zone. Because the initial filtration was indiscriminate, the filtrate still contains vital nutrients, water, and ions. The PCT reabsorbs them back into the surrounding tissue. The remaining fluid leaves the cortex and plunges downward into the medullary pyramid zone, forming a long U-shape known as the loop of Henle. The depth of this loop into the medulla determines the concentration of the final product. By utilizing the surrounding salt gradients mid-deep tissue, the loop extracts remaining water, concentrating the waste. The concentrated fluid ascends back into the cortex, entering a final twisting section called the distal convoluted tubule, or DCT. The DCT acts as a chemical fine-tuner. It regulates specific ion balances and actively manages systemic blood pH before finalizing the fluid. Through this sequential tubular architecture, a crude, toxic biological spill is transformed into a chemically balanced waste product. There is an entirely separate layer of operational complexity occurring simultaneously with this fluid refinement. This schematic shows how the nephron architecture functions concurrently as an embedded endocrine sensor network. Specialized cells monitor systemic blood pressure as it enters the glomerulus. If the pressure drops too low to maintain mechanical filtration, these cells secrete the hormone renin to trigger pressure corrections. The organ also monitors systemic oxygenation levels in the blood. When oxygen dips, it releases erythropoietin, signaling bone marrow to stimulate red blood cell production. Additionally, the cells synthesize calcitriol, the active form of vitamin D required to regulate systemic calcium. These functions establish the kidney as a central biochemical command node, dictating total bodily equilibrium based on real-time fluid data. With processing complete, we can view the finalized divergent outputs of this microscopic sorting. This diagram illustrates a secondary vascular network, the peritubular capillaries, wrapping around the tubules. These blue lines represent deoxygenated filtered blood, collecting the reclaimed water and vital nutrients. This chemically balanced blood converges into larger vessels, exiting the organ through the hilum via the renal vein. Meanwhile, the finalized nitrogenous waste passes from the DCT into a rigid collecting duct, descending back through the medulla. The urine drips from the tips of the medullary pyramids, pooling into cup-like collecting structures called the minor and major calyces. From the calyces, the fluid accumulates into the renal pelvis, a funnel-shaped cavity occupying the center of the hilum. Muscular contractions channel this waste fluid out of the organ via the ureter, directing it toward the bladder for elimination. The anatomical structure of the kidney provides a mechanical solution to a complex chemical problem. It relies on a sequential alignment of macrozones and microtubular dynamics to maintain the chemical boundaries of human life.